Major depressive disorder affects nearly 280 million people globally. Current psychotherapy and pharmacotherapy interventions fail to prevent or resolve roughly half of this illness load. Clinical data suggests that only 1 in 3 mood disorders responds well to traditional treatment. A 2025 systematic review, published in the European Medical Journal, led by Karim and colleagues, examined the dietary outcomes of nearly 80,000 adult participants. This chart plots the upward trends of global ultra-processed food consumption against the rising incidence of major depressive disorder. The data reveals a consistent positive association. Individuals in the highest category of ultra-processed food intake demonstrate a higher risk of developing depressive symptoms. The hazard ratio for this cohort ranges from 1.22 to 1.52. This signifies a 20 to 50% increase in risk, a statistically significant correlation that persists even after adjusting for potential confounders. These findings indicate that diet is not merely a wellness factor, but a physiological determinant of psychiatric health. Understanding this risk requires a shift from traditional psychiatric models toward the microbiota-gut-brain axis, a tangible, bidirectional communication system. The gut and the brain are physically and chemically linked through 3 distinct highways: the vagus nerve, the circulation of immune cytokines, and the endocrine pathways of the stress response. The gut microbiome is a biochemical factory. It facilitates the manufacture of precursors for the body's mood-regulating neurotransmitters, including serotonin and GABA. Because of this direct biological link, clinical depression can have its origins in the lumen of the gut before symptoms manifest in the brain. To isolate the dietary cause, researchers use the NOVA classification system, which categorizes food based on its level of industrial processing. Group 4 defines ultra-processed foods. These are industrial formulations made primarily from substances extracted from foods, like oils, fats, and sugars, combined with additives. These products are engineered for hyperpalatability, using cosmetic additives, artificial sweeteners, and industrial emulsifiers, ingredients with little to no domestic culinary use. Unlike whole foods, these formulations are stripped of structural fiber and micronutrients. They introduce synthetic agents, such as emulsifiers, into a delicate microbial ecosystem. Ultra-processed foods act as biochemical agents that can disrupt the internal environment of the gut. In a healthy state, epithelial cells and tight junctions create a selective intestinal barrier. A diverse microbiome ferments fiber to produce short-chain fatty acids, maintaining this integrity. Specialized enteroendocrine cells then synthesize precursors like serotonin, communicating directly with the brain through the vagus nerve. The stability of emotional and cognitive processes is sustained by the mechanical and chemical integrity of this enteric factory. This integrity is compromised when the lumen is flooded with ultra-processed ingredients and artificial sweeteners while being deprived of fiber. Depriving the microbiome of fiber causes a die-off of beneficial bacteria. This loss of diversity is called dysbiosis, where pathological bacteria begin to dominate. Industrial emulsifiers actively degrade the protective mucus layer and weaken tight junctions. As shown here, this disrupts the intestinal barrier. This leaky gut allows lipopolysaccharide endotoxins to escape into the bloodstream, prompting immune cells to release inflammatory cytokines through systemic immune pathways. Chronic consumption of these foods effectively breaches the body's internal containment, escalating a digestive issue into a systemic immune crisis. These cytokines eventually reach the brain, increasing the permeability of the blood-brain barrier. This breach activates resident immune cells, microglia, leading to neuroinflammation and a subsequent decrease in neurotrophic factors and neurotransmitters. Inflammation also disrupts tryptophan metabolism. Instead of producing serotonin to regulate mood, the body shifts production to kynurenine, which has neurotoxic effects. The brain registers the systemic inflammation as a chronic stressor, hyperactivating the hypothalamus, pituitary adrenal, or HPA axis. This results in a sustained elevation of cortisol. Chronic high cortisol levels are toxic to the hippocampus, physically reducing its volume, a change closely associated with impaired mood regulation and cognitive decline. This biochemical cascade, originating with dysbiosis and ending in neurotransmitter depletion and hippocampal atrophy, provides a mechanistic framework for how diet contributes to the development of depression. Because the gut-brain axis is a bidirectional, dynamic system, these neurochemical pathways are responsive to dietary intervention. Nutritional psychiatry focuses on replacing pro-inflammatory diets with patterns like the Mediterranean diet, which provides a restorative influx of phytochemicals, vitamins, and fiber. Flooding the lumen with fiber and omega-3 fatty acids starves the harmful bacteria, allowing beneficial microbial diversity to recover. The production of short-chain fatty acids resumes, repairing the intestinal wall and sealing the barrier against endotoxin leakage. As the gut heals, systemic inflammation subsides. This allows the HPA axis to normalize and enables the brain to resume the synthesis of serotonin. Nutritional psychiatry is an evidence-based medical approach designed to rebuild the biological hardware of human mental health.